organisms in these tubes as they are the enrichment mediums. These tubes, they are further incubated at 37 degrees centigrade for 24 hours, and then on the next day the growth of the microorganisms is studied. Going towards the clinical significance of the blood. As I said before, blood can acquire variety of different types of infection as it circulates throughout the body. So usually when you go to the doctor suggests to go for the blood test because it is involved in variety of disease. So due to this reason, the blood is one of the most prominent, you can say, the sample which is been tested. Other than that, there are variety of blood parameters that can be studied, like hematological parameters, microbiological parameters, and biomedical, or you can say biochemical analysis parameters. Other thing is that to study the different microorganism growing inside the blood, that is in the blood culture technique. The references that we have taken to prepare our lecture is the textbook of Medical Laboratory Technology by Praful Godkar, Medical Laboratory Science by Ochai and Kulhatkar, Medical Laboratory Practices in Tropical Countries by Cheesebro, and some internet search engines. We hope that you have obtained a very important points from our lecture, and that would helpful to you to study in your further course. So by this we end our lecture. Thank you.
All the best from both of us.
Thank you.
SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.